Welcome to the Molecular Therapy Podcast. I'm Dr. Rory Bricker Anthony, Scientific Editor of the Molecular Therapy family of journals. This episode features a conversation between the section editor of the Oligonucleotide Therapies and Applications section of Molecular Therapy Nucleic Acids, Dr. Marcin Kortieski, and Dr. Norman Michael Drzeniek and Niklas Kotzian from the Berlin Institute of Health at Charité. They will discuss an article recently published in Molecular Therapy Nucleic Acids titled mRNA-based CAR T-cell engineering. Unmodified mRNA enables high CAR expression without innate immune activation in T cells. But first, the window is officially open for late-breaking abstract submissions to the ASGCT 2026 annual meeting, happening May 11th through the 15th in Boston. If you have high-impact, newsworthy data that was not available during our original abstract submission period, now is your chance to submit your work. You can help ensure the most current and transformative science is represented at this year's annual meeting. Head to annualmeeting.asgct.org/abstracts to get all the details and submit by March 13th. And now, Dr. Kortieski, Drzeniek, and Kotzian.
Welcome to the Molecular Therapy Podcast. I'm Marcin Kortieski. I'm a section editor for Molecular Therapy Nucleic Acids. And today we'll talk with a team whose new study that appeared as a brief report in Molecular Therapy suggests that we may not need modified nucleosides for mRNA-based CAR T-cell engineering. So this finding could simplify manufacturing and reduce costs. So we have with us Dr. Norman Drzeniek, the corresponding author, physician scientist from the Department of Rheumatology and Immunology at Charité Medical University in Berlin, Germany, and the co-first author, Dr. Niklas Kotzian. So welcome, and if you can briefly introduce yourself.
With pleasure. Thank you for having us on the podcast. My name is Norman Drzeniek. I am a physician scientist and bioengineer based in Berlin at Charité Rheumatology and at Berlin Institute of Health, in the group of experimental immunotherapy, where I lead a small team focused on nanomedicines for autoimmunity. Right now, I'm a visiting research scholar with Dr. Mike Mitchell at the University of Pennsylvania. And my primary research interests and what our team is focusing on basically is engineering and advancing technologies for targeted immune modulation and targeted treatment of autoimmune diseases. We're interested also in fibrosis and aging.
Excellent. So that's pretty broad background. So, Dr. Kotzian?
Yeah, sadly not a doctor yet. I'm a student at the university right now still. I'm studying biochemistry at the FU Berlin. And I joined Norman on one of my internship for my university degree, actually, because I was interested in immunology and also biomedicine in general. And I learned a lot about RNA, and we did this project together. I think we have great results in this publication. And now at this moment, I moved on. I do an internship in multiomics right now, and I want to move on in aging long term, I think, in aging science. I also always have LMPs back in my mind, and I'm thinking about how I could apply them in medical applications in the future as well.
Yeah, so that's pretty interdisciplinary background too, bring you to biomaterials, and that's wonderful. So let's start with getting some background on your research. So for many years, as I think many people know, the unmodified in vitro transcribed mRNA has been seen as highly immunogenic. It can trigger different antiviral sensors, interferons, inflammatory pathways, or even apoptosis. So that's why how the modifications like N1 methylpseudouridine or methoxyuridine became a gold standard. And of course, they brought also Nobel Prize awards to Drs. Katalin Kariko and Drew Weissman. So for listeners who may be less familiar with this topic, can you give them a brief primer on what the innate immune sensors for nucleic acids are? And why does immune activation matters for CAR T cell production?
Yes, of course. As you summarized, basically this research into nucleic acid innate sensing has been a very active field in the past years. So cells are equipped with mechanisms to recognize DNA and RNA, which is not their own, so exogenous nucleic acid, because it's a virus sensing mechanism, right? So when a virus infects a cell, it introduces its nucleic acids to reproduce. So all our cells have basically mechanisms to recognize that and shut down their protein production and undergo basically programmed cell death or cellular suicide, if you will, to prevent reproduction of viruses. And this has been a huge hindrance in, of course, introducing synthetic nucleic acids into cells for therapeutic purposes. So this has traditionally included classic DNA-based gene therapy. And then in the late 90s, early 2000s, people were exploring more and more mRNA as an alternative vector for, well, not gene therapy, because it isn't gene therapy, but it's a template that can encode any protein you want to produce in the cell. And this has been explored with the idea, for example, to overexpress a chimeric antigen receptor in T cells or for protein replacement therapy in genetic defects. But the idea, the biggest challenge turned out to be actually this antiviral immune response that cells seem to have, which made it very difficult to introduce nucleic acids into cells without a penalty, right?
Mm-hmm. Yeah, I think that's a good wrap up. I will just add as a side note that this innate immunity can also be used for beneficial purposes. That's what we use in generation of vaccines. So toll-like receptor 3, 7, 8, or TLR9, a CpG agonist that, I mean, they are quite useful, but it has to be done in a controlled manner. Immune activation can get out of hand. So your previous research focused on effects of stromal cells and the response of stromal cells to unmodified mRNA that generated inflammatory responses. So it was how this project started. Was that related to your previous work?
Yes. So originally in my PhD, I started, and this was before mRNA became such a prominent topic. We had the idea of using mRNA to encode cytokines and express them in stromal cells. To essentially, the problem with cytokine therapy is often the short half-life of these agents. So our idea was to use mRNA to extend the therapeutic duration of cytokine expression, and to possibly transplant mRNA-modified, mRNA-transfected cells that would produce these cytokines locally as a sort of living drug factory. And this was an idea early in my PhD, where the barrier was to do that without creating inflammation coming from the antiviral response against the mRNA. And we presented these results a few years ago at a conference, and we got a lot of interest, but also a lot of questions of people looking at our bone marrow stromal cell data and asking, hey, I work with macrophages. Would this be applicable to my research? Or, I'm trying to overexpress something in endothelial cells. What would that do? What would that look like? Would you expect the same immune responses? And would the mitigation strategies that you propose also work for my application? And so we realized that there are very few studies that actually bridge, look at this innate immune response toward nucleic acids in a more application agnostic manner and compare the sensitivity of different cells.
Mhm. Yeah, I think if we can look into your paper to find a really nice screen of different cell lineages, including stromal cell, epithelial cells, of course, macrophages, which are an excellent source of sensitivity to nucleic acids in general. But in your screen, T cells were totally different with no spike in interferon, no increased cell death or comparable expression across unmodified, modified mRNA. So was it surprising to you in any way?
Yes,
Of course.
absolutely. The study started out completely without T cells initially. So we started out, we had a lot of data from previous project on the stromal cells. We were interested in autoimmune diseases, potentially looking more at fibrotic tissues or endothelial cells, macrophages, of course. We knew from other projects that the doses we had optimized for all these different cell types in individual projects were different. So we were screening these different uridine modified mRNAs across those different cell types, and around the same time, a master student who I was co-mentoring at the time was interested in trying lipid nanoparticles for T cell transfection. And I told her, try to compare mRNAs with different nucleotide chemistries. Let's see if this looks similar to what we see in macrophages or other cell types. And she came back with this result, GFP under the microscope, and all the cell conditions, regardless of which uridine chemistry she used, were equally bright. And this was incredible to see, because in the macrophages, we would get very high and very bright GFP signal from N1-methyl pseudouridine, from 5-methoxyuridine, but only very dim signal from unmodified uridine, and also a lot of cell death from the unmodified mRNA. And in T cells, everything looked the same. So, I asked Nurhan, one of the first authors of the study we're discussing today, who was actually doing this cell type comparison. I asked Nurhan, can you include T cells and have a look at that as well, and include that possibly in your screening? And we were very surprised to see that actually the T cells, as you mentioned, didn't really show any measurable type 1 interferon response or cell death, even toward unmodified mRNA. Completely unexpected. Yeah.
Yeah, I think one of the controls we were doing. Go ahead, Dr. Kotzian.
I wanted to just say that it is especially interesting because T cells are, of course, one of the most interesting cells in regenerative therapies right now, I would say, with CAR T cells. So there would be a lot of application for this, of course. This is also one of the reasons why we really looked closer at it on this paper, yeah.
Yeah, and what I found interesting also, you double checked whether delivery method does not influence the response of the cells. T cells are far less likely to internalize LNPs, or they are more difficult to definitely transfect. But with electroporation, you have basically seen, I think, similar effects, or were there any differences? Because this is far more kind of drastic way of handling the cells.
Yeah, we saw that the effect was independent of electroporation or using LMPs. So we saw the same effect as to the same effect on the RNA response in the macrophages across electroporation and LMPs, which would be a high response, and the same low response of T cells against electroporation and lipid nanoparticle transfection. So the effect was really an RNA effect we saw here, independent of the delivery method.
Mm-hmm. Yeah, and you analyze the public RNA-seq data, and finding that basically TLR7, TLR8, which are both uridine sensing receptors, were expressed in fact in lower levels in T cells than in other immune cells that you tested. At the same time, T cells are not deprived of RNA sensors, or they do express RIG1 or MDA5 and so on, as shown in your data. So, how far do you think these findings go towards explaining basically the effect that you have observed?
Yeah,
Yeah, this is a very important point. It's important, and we also stress that in the paper that at the present time we didn't go in detail in elucidating which of these exact sensors mediates, or in the case of T cells, doesn't mediate the innate immune response that we expected to see. So this is only indirect mechanistic evidence where we see this clear difference. We know that TLR7 and 8 are typically thought of as the receptors that recognize unmodified uridine motifs on the RNA. And serendipitously, we realized that these are expressed, either not expressed or expressed only at very low levels in T cells, versus, for example, macrophages, which express a lot of TLR7 and 8. So this is, a correlation that doesn't prove causation, of course.
Yeah, that's a good point. I think let's move maybe to basically the CAR T cell application to CAR T cell expression levels. So your finding is showing quite clearly that the CAR Ts engineered with unmodified CAR mRNA showed higher expression of the chimeric receptor with comparable viability and also quite normal cytokine secretion profile. So this is quite striking, given how essential most of the groups think that keeping mRNA unmodified is standard. So why do you think this unmodified mRNA translates better in T cells? Is it a sequence-dependent effect? Would that be related to interactions of modified nucleotides with ribosomes inside the target cell?
So this is one thing that would be really interesting to go and further study in the future. There are, of course, different things one could think of, right? So using unmodified uridine would maybe lead to a different secondary structure of the RNA or using unmodified uridine maybe also is beneficial for binding of different proteins in the cell. So there are different kinds of approaches that could be responsible for this effect that we are seeing, and not only we are seeing, but also other groups are seeing.
Mm-hmm.
Very true. I want to mention here as well, several groups have actually read the paper and contacted us saying, hey, we also saw this, but we didn't thought of reporting it.
Yeah, that's a common problem of data getting lost in the drawer. So what are the implications in terms of T cell engineering? With that finding, have implications also DNA-based engineering? You can use mRNA, you could also use plasmids and so on.
So I think this is a very far-reaching question. We focused in this study on CAR T cells because this very discussion, do we use DNA to produce CAR T cells? Do we use mRNA? And if yes, do we use it ex vivo? Do we use it in vivo as in vivo CAR? That's a very prominent ongoing discussion in the CAR T cell community. So we chose that. We are interested in that application, and we chose also to showcase our finding, because actually colleagues from Charité, from the team of Dimitrios Wagner, investigated a few years ago CRISPR-Cas9 modification of T cells or engineering of CAR T cells based on CRISPR. And they found that actually this cellular immune activation toward the DNA HDR template they were introducing for CAR knock-in was limiting the efficacy of their cell engineering. So at some point they couldn't introduce more DNA into the T cells because the T cells would respond with sting-based,
Activation.
stimulated immune activation, which would then in turn limit the expression again, right? So you have this very narrow therapeutic window where you can't dose more RNA or more CRISPR knock-in DNA template because the cell just, arms an immune response toward it. And with mRNA, we don't see that. So in terms of implications of our study for immunotherapy, we are developing these mRNA-based CAR T-cell therapies for autoimmune disease with the idea of generating an immune reset in patients with severe systemic autoimmunity without having to use virus-modified CAR T-cells. And mRNA CAR T-cells are becoming a promising alternative, also a safer and potentially more cost-effective alternative to traditional virus-based CAR T-cells. So I would be excited to see more research in that space as well. And that's definitely something that we are working on as well.
Excellent. Yeah, I think it was a good point to make. It's not about CAR T only in oncology.
Right.
Use right now. Huh?
I think also in oncology, mRNA-based CAR T cells have been explored a while ago, but didn't result in very robust responses. And it could be that in the oncological setting, you need the stable CAR-expressing T cells. There is actually a meta-analysis which shows that CAR T persistence correlates with efficacy in oncological patients. And in autoimmunity, we don't know yet because we see the CAR T cells are disappearing anyways once they kill the B cells. So it could very well be that a transient CAR T effect is enough to reach a stable drug-free remission in autoimmunity.
Yeah, so I think what comes out quite strong, and you noted in the paper, is this reduction production cost for mRNA-based CAR T, which is roughly, I think, 35%. That's what you write. So that could really impact the CAR T workflow. But just to be clear to listeners, there is a catch. The unmodified mRNA would not be necessarily safe if you would use it for generation of in vivo or in situ CAR T cells. So, which are right now intensely studied because they could bring down the cost and kind of open up this technology to many more available medical centers. But there is a risk here that off-target cells, different immune cells, macrophages, could be taking up the LNP as well. So do you think any safeguards that could be applied here? And at what point and what part of the system?
Yeah, so I think, of course, we have to carefully interpret these findings when talking about in vivo CAR T, right? So when we deliver the mRNA via nanoparticles systemically and we try to target the T cells, that targeting is never going to be 100% selective. That's something that is, a very prominent research effort right now to improve cell-specific targeting of lipid nanoparticle delivery. But essentially, our findings are significant for that field in the sense that if you know that immune responses are cell type dependent, then essentially mitigating immunotoxicity becomes a biodistribution problem. Meaning that if you know that the macrophages, for example, as our study shows, are the most sensitive cell type, exhibiting the strongest immune reaction toward RNA, then you could think of a strategy that, for example, detargets the macrophages and tries to target T cells as much as possible to mitigate also a systemic immune response, right?
Yeah, so hopefully we see development of more and more specific LNPs, but that's right now that's of course an ongoing battle.
Yes.
Oh yeah, so what's next? I think in your plans. I mean, you mentioned that there is some open space and open questions left about the mechanistic effects of RNA sensing in T cells, which would be really interesting to explore, I think, because that really could generate more impactful papers in the future as well. So what experiments you see as coming up next?
Yeah, generally, it's really this mechanism that is still interesting, right? So we see that different cell types have a different response to the RNA. But we also see that T cells still have some RNA receptors that are expressed and some pathways that are expressed. And now the question arises, why don't the T cells respond to the RNA even though they have these mechanisms still active, which we can see, of course, in the genomics data, right?
Hmm.
So, for example, many of these receptors are targeted towards double-stranded RNA, and this may be a limitation of us using uridine-based RNA. Yeah.
Yeah. So the our study doesn't clearly investigate how the RNA is sensed. There's a lot of literature showing different motives on different classes of nucleic acids, including the cap, the nucleotides themselves, or the double-stranded structures, which are all sensed by different receptors. And each of those could also be cell type specific, right? And there are more and more papers coming out. Every now and then you have a new paper showing a new non-canonical role for one of those receptors,
Hmm.
or a response that hasn't been previously recognized. So it's important to stress that this is still an active research field, which is not at the forefront of our focus. We are more translational and application oriented toward
Hmm.
immunotherapy. But I think there's an incredible potential in fully understanding the immune sensing of nucleic acids, also at this level of cell type dependence, and also the immune responses, for example, toward nanoparticle components, are also completely unclear. So there's a lot of mechanistic insight I expect in the near future to be coming there. In terms of our own study, I think it would have been cool to overexpress toll-like receptor 7 and 8 in T cells and see if we can restore the sensing, or if there are other pathways in the signaling chain, or parts in the signaling chain which are also hindering the immune response in T cells.
Yeah, that definitely exciting next steps. And also, I think we mostly focus on engineering kind of biomaterial kind of point of view. But I think one interesting possibility that those studies pointing to is also that this observation creates an opportunity for immune modulation to naturally occur in the tumor microenvironment. So if T cells can tolerate and even utilize 4' mRNA, exogenous mRNA, they could easily also utilize mRNA coming from other cells. So they could be positively or negatively regulated by mRNA transfer. We know about the transfer happening using extracellular vesicles or exosomes. But I don't think anybody has reported or looked into whether T cells would be a target as well. I think, mostly focus on endothelial cells or macrophages, monocytes. So I think that would be probably interesting biology behind it as well, beyond just a practical point of view on that your studies is
Right.
really strong at. I think we are getting close to the end of our conversation. So would you have any final takeaways or statements that you would like to do?
I think one thing that we haven't mentioned yet
Mhm.
is that actually one of our collaborators has seen a similar finding in NK cells. They have also, I think it's actually also published in Molecular Therapy Nucleic Acids, where they observed that unmodified mRNA outperformed modified mRNA in NK cells.
Mm-hmm.
So I think it's worthwhile to look at these sensing mechanisms in different primary cell types. I think a big limitation in the field is that people use cell lines, which do not necessarily reflect this biological variability in those sensing mechanisms.
There's definitely a tendency to follow certain path without questioning again, if despite of having new reagents, new delivery methods, and additional ways to protect oligonucleotides or mRNA.
Yeah, I guess that's often the case. Also, our study, I think, shows that uridine, at least for our CD19 CAR, outperformed the 5-methoxy and especially the one pseudo uridine. And it would be interesting to see, because this would be interesting, of course, for applications later on, if this is true for other CD19 cars as well, or if this is just a sequence-specific effect, right? And I think this is an important thing to figure out going onwards.
Yes, I agree for sure.
That's true. We have actually in the supplement data on a second CAR where the expression levels of the unmodified mRNA were similarly high. So there seems to be a lot of consistency there, but the expression level of N1-methyl pseudouridine was low for one of the CARs and high for the other. So it seems that, the story doesn't end here. And I would encourage everyone to compare some of those modifications for the very mRNA that they're trying to express.
All right. So thank you, Dr. Drzeniek, Mr. Kotzian, for joining us today. Congratulations on your impactful work. Thank you to our listeners for tuning in, and see you next time on the Molecular Therapy Podcast. Thank you.
Thank you.
Thank you for having us.